welcome to the webinar series 3.0 organized by JIS Institute of Advanced Studies and Research, Kolkata. I'm Devabani Ganguly, today's host. Before I introduce our speaker, Dr. Anuradha Ghosh, let me show you a short video to introduce ourselves, that means JIS Institute of Advanced Studies and Research, Kolkata.
A vision emerges in the mastermind of Padma Shri Professor Ajay Kumar Ray to develop a unique postgraduate research institute with a difference, promoting an interdisciplinary approach, offering translational research-based education, and focusing on areas with high-quality job prospects and entrepreneurial opportunities. Fortunately, JIS Group educational initiatives joined hands in this dream project. And then, in mid-2019, under the aegis of JIS University, JIS Institute of Advanced Studies and Research Kolkata was born, with 3 centers: Center for Data Science, CDS, Center for Health Science and Technology, CHEST, and Center for Interdisciplinary Sciences, CIS. Now, let's dive deep into the CHEST. The demand of technological expertise in both academic and industry sectors of health science can only be ever increasing. On one hand, there's a continuous need for biotechnological developments, especially for direct medical applications. Simultaneously, the advent of newer high-throughput technologies asks for powerful bioinformatics solutions on how to extract, tackle, analyze, and visualize the biologically meaningful data for subsequent research and industrial applications. This scenario puts qualified bioinformatics professionals, especially the ones with expertise of analyzing medical biotechnology data, are in high demand for top salary jobs worldwide. Keeping this in mind, CHEST offers 2-year MSc in Medical Biotechnology and Bioinformatics, 2-year MTech in Bioinformatics, and regular as well as integrated master's PhD program in related fields. But why would you choose CHEST? Here are 3 reasons why CHEST is the best nest to hatch your career flight in health and medicine. Number 1. State-of-the-art molecular genomics, biotechnology, and bioinformatics laboratory with avant-garde equipment like PCR, real-time PCR, tape station, gel documentation system, CO2 incubator, fluorescence and UV spectrometer, biosafety cabinet, fume hood, and many more, along with ample laboratory workspace. And finally, last but not least, Illumina MiSeq, one of the highest ranked platforms for genome sequencing. JIS IASR boasts to be the only private educational institute and one of the very few among all public plus private institutes in Eastern India to have the in-house facility of this high throughput next generation sequencing and data analysis. Number 2. Internationally experienced faculty team with prestigious research grants, top-tier journal publications, and global collaborations. Multifaceted domain knowledge of CHEST faculty members aims a paradigm shift in infectious diseases research in India. The expertise in the CHEST faculty team spans from molecular microbiology and post-pathogen interaction, adaptation strategies of microbial pathogens, evolution of microbial virulence and antibiotic resistance, molecular modeling to explore functional insights of biomolecules, to as diverse fields as image processing and machine learning, as reflected via seminal publications in top-tier journals like Cell, Nature, Nature Light Science and Applications, Nature Protocols, Nature Communications, Journal of the American Chemical Society, PLOS Computational Biology, Proceedings of the National Academy of Sciences USA, to name a few. This has been possible through worldwide collaborations with academia and industry, and prestigious research grant funding from Government of India and Wellcome Trust in United Kingdom, an unparalleled achievement of an Indian institute within the first year of its inception. Number 3. Need of the hour biotechnology and bioinformatics programs in health and medicine. Along with a solid foundation of different aspects of medical biotechnology, computational biology, and bioinformatics applications, our MSc and MTech programs include some of the key subjects of utmost industrial needs: For example, next-generation sequencing data analysis, microbial virulence and antibiotic resistance, proteomics and metabolomics, computer-aided drug design, pharmacoinformatics, systems biology and biological networks, big data in bioinformatics, and so on. And expertise in any of these fields can readily offer lucrative jobs in pharmaceutical and biotechnology industries, in hospitals and clinics, in bioinformatic data analysis companies in India and abroad, besides the standing opportunities of research and academic positions. Apart from regular subject-oriented topics, our programs consist of topics like research methodology, career development in entrepreneurship, and intellectual property rights. This edition not only enables the students to cope with the situations in industrial or academic environments during their summer internships or final semester projects, or in their job positions after the course completion, but also aims to motivate the students to develop an entrepreneurial mindset. So, why delay? Join CHEST, the best nest to hatch your career flight in health and medicine, and target the top together.
So I'm privileged to introduce our today's speaker, Dr. Anuradha Ghosh, one of the experienced scientists in the field of microbiology. Dr. Ghosh completed her PhD in environmental microbiology and bacterial diversity taxonomy from Institute of Microbial Technology, IASR Institute of Chandigarh, India, registered from JNU, India in 2008, right? She was a postdoctoral fellow of Department of Diagnostic Medicine and Pathobiology at Kansas State University, Manhattan, Kansas in USA from 2008 to 2013. In the same department, she was appointed as research assistant professor and served till 2015. Then Dr. Ghosh has moved to Department of Biology in Pittsburg State University in Kansas. She is working as associate professor, my mistake, and serves as PSU master advisor, lead pre-vet, chiropractic. I'm sorry, I don't know the term. My mistake. My apology. Pre-optometry advisor, pre-med, dental advisor, and many such positions too big to list. Her research interest comprises of environmental microbiology, which includes antimicrobial resistance genes in hospital environment, bioremediation, vector-borne disease. She is also interested in food safety with respect to ASTAC and enterococci, microbial ecology, and bacterial systematics. Also, her interest in molecular diagnostics, which includes genotyping and whole genome sequencing. She is awardee of so many prestigious awards, grants, including NSF grants, Walton Fellowships, etc. She has authored and co-authored 34 peer-reviewed articles and many newsletters, book chapters. So on behalf of JISR Kolkata, I convey my sincere thanks to Dr. Anuradha Ghosh for accepting our invitation for this webinar. Dr. Ghosh, now the stage is all yours. Dear attendees, please write your questions in Q&A section or chat section. Our speaker will address all the questions only after the talk. Thank you very much, Anuradha, once again. Please, you can now start sharing. Thank you.
Thank you, Dr. Ganguly, for your kind words of introduction. It's really my privilege to be here as a guest speaker in your prestigious seminar series. And I think I'll be talking to such a laureate attendees. Some of the basic stuff that I thought of explaining, probably I'm not going to do that because all the bioinformaticians and whole genome sequencing experiences, most of you have that already. I'm going to share my slide first and then move on to the talk. Okay, I hope everybody can see these slides. As you see, like I want to talk about bioinformatics application in microbiological research. I have been trained as a microbiologist, so this is something new I acquired after I joined here at Pittsburg State University. So this is one of the areas I'm still like learning and trying to gain enough experience so that I can move on using bioinformatic approaches in several different fields. I did have a slide showing my introduction, but Dr. Ganguly, I think, gave the introduction pretty well. I don't have to add much here. I'm born and raised in Kolkata, and I did my BSc and MSc from the same place. And then I moved to Chandigarh to do my PhD. And after that, I landed on Kansas. And here I did my postdoctoral in Manhattan. And then I moved to Pittsburg State University as to join the graduate faculty as an assistant professor in 2015. I want to give a little bit background about our university. This is not a primarily research university, so it is not a like first tier research. It's mainly undergrad teaching university. So we have different responsibilities. So some of you may not be aware of this kind of four-year universities where teaching is the primary thing that you're supposed to do. We have a very high teaching load. Year around or during the academic year, we are supposed to teach 22 to 24 credit hours, and that leaves us with at least like 4 to 5 different courses during the semester. And then we have a very unique advising system. Advising means you have to take care of the students, what classes they are going to enroll every semester. You have to meet with them individually. And we have like assigned advices up to 30 different advices every semester. So this advising is the second priority after the teaching. And in our department, we have almost like 90% of the students who wants to become a healthcare professional. There are different areas of healthcare, and that's what Dr. Ganguly was explaining. There are pre-med and dental. So we prepare the students, try to like complete all the prerequisites so that they can apply for medical school after 4 years. And then we have students who wants to go to chiropractic or optometry schools or want to become veterinarian. So all these prerequisites we have to actually complete during these 4 years. And it's a big responsibility. And then comes research. So now you can see like research is not really a priority. So in a way it is good, like it is not like a research in universities where if you do not write a grant or if you cannot get an NIH grant, your job is at stake. It's not like that. As long as we are teaching very well, we are having good student evaluation, and the success rate in admission of the students in those professional schools are good, we are good to go. But since all the faculties in the department, we had a lot of like research experience in the past, so we are encouraged to retain our scholarship and do some research which is going to be beneficial not only to the faculty but to the undergrad and graduate students. So we have a graduate school in Pittsburg State, which is, again, if you compare with some other state universities which are primarily teaching, they may not have the graduate schools. In biology, we have a master's program. So it is not a PhD program, it's just a 2-year master program. So we get students in that program. And we do have a NIH-sponsored program called K-INBRE, and that's an immense support to our system because they provide several scholarships you have to apply, and they promote a lot of like undergrad research. With this, the outline of my talk today is to go over the theme of our research lab. In the introduction, you heard about different research interests. So I would like to show you like how these different research interests can intricate together. And then why I want to apply bioinformatics in our research projects. And in the end, I'm going to just explain briefly about the application of bioinformatics in 2 of our recent projects that we just completed. One in detail, the other one I'll just briefly touch. So the theme of our research is One Health. If you type in what is One Health, you'll see a list of hits coming up. There are One Health related conferences also. There is a One Health initiative that is like internationally recognized. So how this concept developed? So we know like the population, human population is growing and expanding, and they are reaching to new geographical areas. And that way you're coming in very close contact with the domestic and wild animals. And this close contact increases the opportunity to spread some unknown diseases. And we are in the era of coronavirus, so all of you are aware of like how the diseases or the viruses that are native and asymptomatic to the animals can create problems in other living beings, including humans. And also with this growing population, the movement of people, animals, animal products, they have also increased a lot because of international trade and traveling. So we do not have any like barriers of borders. The crossing the borders you can do in minutes, in hours. And that's why if we try to only focus on the well-being of human health, it may not be possible if we are not going to consider the environmental health and animal health. Now this term, like One Health, came in like early 21st century, but One Medicine, this term was coined long before, like in 1916s. It was coined by a veterinarian, and he tried to show like the medicines that we use for veterinary practice has got significant overlap with the human health or human medicines that are used in human treatment. So he introduced the term One Medicine. It was not that noticed at that time, but as we approach and have this population blast, we now realize. So I'll just give you a description what is given by the One Health initiative. It's a collaborative efforts of multiple disciplines working locally, nationally, and globally to obtain, their aim is to obtain the optimal health for people, animal, and environment. Now there are several issues that are related to One Health, and in our lab, we the projects that are underway that touch, actually you'll see several of these issues. I don't want you to just go over it. I'll describe it in my next slide. So the undergrad and graduate research that are going on in the lab, some focus on environmental contamination. So it is directly related to environmental health. So we have Pittsburgh. The name came from Pitts. So there is another Pittsburgh in Pennsylvania. So these areas were mined for a long time because of a lot of coal around this area. And the coal mining was done by mainly surface mining, which means you remove all those rocks from the surface, and either you make like create strips or pits, and then you take out the coal. So this is basically an abundant mining town, but it flourished later because of the development of university and other industries. So some of the projects will look into this abundant coal mine land, so how they're reclaimed. And a little bit south from this southeast Kansas, there is another state called Oklahoma, and where in the like over 100 years, there have been a lot of mining for lead. So we found called Tar Creek, that is a lead contaminated site. So after mining, they didn't reclaim one. So another project in the lab, we focus on this lead contamination. So this part touches a little bit about the environmental contaminations. So our approach is bioremediation, means using microbes, how we can take care of those sites or clean up those sites. The other area that we are looking into, and this is mainly based on my experience in my postdoc, I worked with several vector-borne pathogens and vectors as such. So in this particular area of cancers, again, there are many vectors, and one of the primary vector is tick. You may be all aware of dog ticks. If you have a pet at home, you take care of the ticks. But here we have different types of ticks, not only dog tick, it's wood tick, deer tick. By the way, in this area, we have a significant amount of woods, so deers are in abundance, and they carry these ticks, and there are diseases like Lyme disease, ehrlichiosis, Rocky Mountain spotted fever, tularemia. These are the ones that are carried by the ticks. So when I moved in here, in this part of Kansas, it gave me some opportunity to work with this kind of vectors. Another one is mosquitoes. West Nile virus is the virus that is abundant here, and that is carried by Culex mosquitoes. We fortunately do not have many other species which carry any other disease here. So we have some surveillance projects going on in this respect. In One Health, we talk about food safety, and another project, it is a collaborative project with University of Arkansas Food Science Department, where we are looking at poultry feed. So chickens, of course, we are going to eat them, but what the chicken eat, that matters also, because eventually that is going to stay in their gut. So this is one part of the food safety, which sometimes the farmers ignore. But the FDA and USDA, they put a lot of emphasis. So there is a lot of funding opportunity in this kind of pre-harvest food safety. And one of my students was involved in this project. Antimicrobial resistance, which is, we know this is one of the topmost concern in human health these days. Starting in 2000, early, like, I think 2013, CDC and NIH, they have created separate areas that look into this antimicrobial resistance. Now here I would like to emphasize like another aspect of our university. So it's very easy to find undergrad researchers here who are going to be very motivated to work in the semester, but it is difficult to keep them for a long time to finish the project. And that's why you see so many undergrads here actually involved in the project, but it's still going on. And it's sometimes challenging until you convert that project into a master project and have somebody assigned to work on it 2 years to complete and then lead to a publication. So it's just a thing I would like to share, because as a faculty, we face this kind of problems a lot. With this backdrop of my lab, I'll move on to bioinformatics. So most of the things that you're seeing here, the project topics are somehow related to bacteria. So microbiology work is the primary thing we were doing. And with the surge of like genome sequences in recent days, I thought like this is going to be the future. So why not gain some experience in this field and try to integrate it or implement informatics in our projects. So here is a kind of definition you can say for bioinformatics. We know that it's an interdisciplinary area, and as I know, like I'm talking to most like reputed bioinformaticians, probably they are trained ones, so I don't want to spend too much of time in this. So the job of biology is to acquire data. Okay, and then computer scientists, information technologists, they are going to create all those fascinating tools, softwares. They'll produce all those algorithms. I have seen Dr. Ganguly developing algorithms in the lab, so I know like how tenuous those works are. So they create all those softwares, and then the bioinformaticians, they are going to analyze the data using all those softwares. So they have some biology background, of course, so that they can answer the biological questions. Now, as I said, like I need to undergo a steep like learning curve to understand this bioinformatics. I had a brief introduction to bioinformatics when I was master's students in Rajabhaja Science College. but I never thought of doing bioinformatics on my own. It's not because like I didn't like it, but I probably liked microbiology more than this. Now, in our biology department, it's not all about your research. You have to really see like how your research is going to impact the students. So that's why it was very important to convince my colleagues like this informatics, if we do as a research in the lab and can integrate it with some curriculum, biology curriculum, then it is going to develop a core skill, because students are learning the basic principles of genetics, microbiology, evolution, ecology, all these different fields. So they can really apply the bioinformatics in these fields, and it can be an asset to the students. So that's how we try to convince like this is the future, and as a core skill, if they can gain this after these 4 years of their study in this undergrad university, then they're going to leave the university with this thing. And if they apply for clinical labs, they'll have the advantage because sequencing is one of the primary things that's going on. If they're in PhD program, that could be another like accomplishment for them to get into there. Now, what bioinformatics can do, again, it's more of a teaching slide. Sequence assembly, gene annotation, we know these things. Molecular evaluation, probably it's the right time to talk about it because, again, the coronavirus outbreak, it's a pandemic, so people are gathering like different strains from different part of the world, and then they are trying to see compare the genome sequences and trying to see like what are the mutations that are making one variation to another variation. In 2009, when there was a swine flu H1N1, I think the first time this kind of like molecular approach was taken to understand the course of this outbreak of H1N1. And then there is comparative genomics, functional genomics. Those you can do if you have the genome data available, and then you compare with transcripts and proteins. And this structural genomics, which again, Dr. Ganguly and their group, they're doing it, I guess. This is to look at the structural integrity and then how the protein and the drug and targets interact with each other. So these are some of the booming fields where you can apply. This slide shows mainly like in terms of eukaryotic genome sequences, you see there is a surge. And in 2003, once the Human Genome Project was completed, there was no looking back, basically. There were several advancements in the sequencing techniques, and that led to this like exponential increase in number of sequences that have been deposited and sequenced properly. In terms of eukaryotes, there is the computer scientists or data scientists are aware of the Moore's law. So this graph basically follows Moore's law, and it shows like how that sequence, with the cost of sequencing getting lower, how you can actually populate the sequences in the database. I'd like to just briefly give you an idea, like 2003, the human genome was completed. In 2007, can you guess what was the cost of sequencing a whole genome, human genome? Because this will tell you like how over the time it changed, basically. You can just type in the question in the chat room or somewhere. Okay, I'll give you the answer probably here. Yes, I got one here. It's 10 million, so it's really close to what you said. Thank you. And then in 2011, when there were a few advancements, I remember like I was using pyrosequencing in 2011. That time pyrosequencing was there, and then in few years it got obsolete. In 2011, can you guess what was the cost of human genome? with some introduction of next generation sequencing. So from 10 million, yes, that's also very close. It's actually $10,000 at that time. And then in this era, now we are talking about $1,000 per genome, right? And we are also talking about personalized medicine. So it's really affordable. And that's all because of these advancements made by all those bioinformaticians and computer scientists. And of course, biophysicists who actually work for the development of the sequencing platform as such. So that's how you see that this populated graph is highly exponential graph is because of this lowering cost over the time. This is website backmap that shows like how many prokaryotic genomes have been sequenced so far. It's about 3,000, and this includes archaea and bacteria. So both are there. Of course, you see the difference. It was like around 600 for the eukaryotes and 3,000 for prokaryotes because it's a smaller genome. It's easy to handle and it's easy to sequence. Now, biological databases, they play a very important role in the bioinformatics, because if you have trillions of bases floating around and there is no way to represent all those data in an organized way, then it's not of any use to the scientific community, right? So that's why it's very important to organize them. And the website that you see here, it's sequence-based GenBank, which is from NCBI. We have UCSC Genome Browser, Ensemble. So these are all like sequence-based databases and portals. There are some which are model organism-based, like worm-based. TAIR is for Arabidopsis, and then MGI is related to Mouse Genome Institute. So there are some, like if you are working with specific organism, then you can always look for these and not going to the GenBank maybe to look at a broader database. So they have refined databases, and some of them like there's online Mendelian inheritance in man, OMIM, protein data bank, or encyclopedia of genes and genomes. These are non-sequence centric databases. These are all available. So databases, as you see, like they give that platform to the several scientists, whatever field they're working in, and these are publicly accessible. So it's a wonderful way to access others' work, other sequences, and then you can do all those different genomics based on that. Now, with this background, I'm going to now talk about the projects that we did in the lab. And again, keeping that One Health initiative in mind, the projects were designed, and then eventually we applied bioinformatics to like add another step to that project, you can say. So Rachel was my first master student, and she worked, looked at this abandoned coal mine area, which was kind of a local environmental pollution issue. So it was nice to look at that the very first time. Then we came here and then have some like ongoing long-term project in terms of remediation. And then Brady Stenbock, who was a molecular biologist, but I turned him into a bioinformatician in later stage. So he actually applied whole genome sequencing to that particular project. So let me introduce what is abandoned coal mine land. So as I mentioned, like we had a lot of strip mining or surface mining, which created pits and strips here. So when you do this kind of mining, you're exposing the rocks, and this rocks, the abundant rock that you kind of find in this coal mining area is pyrite. It's a iron disulfide. And when this pyrite is exposed to oxygen and then it also comes in contact with groundwater, they are going to form sulfuric acid. And this in turn is going to lower the pH of the water in that area. And then some form of this iron is going to get precipitated. And you'll see a yellow precipitation around that area. And this is called acid mine drainage because the pH is low, and that's why you have this iron precipitated over there. And that gives this orange color to the water. This is extremely deleterious to the living organisms, aquatic fauna, flora, even the surrounding, they get damaged. And especially if there is residential area close by, then there is chance like your groundwater is also getting contaminated. Because of the color, sometimes the local people call it fool's gold or yellow boy. These are local terms that are used. So we chose a site. This is abundant coal mine, but this is partially reclaimed. In 1970s, there was an act, and a reclamation act was introduced, and the companies who mined and just left the site without any treatment, they were supposed to at least partially remediate those sites. So this area was remediated, but it's partially remediated, so it still had a lot of issues. And this is one of the sites that is owned by Pittsburg State University. So it was like easy access to do research in that area, and that's why that was our priority. But there are many other sites in other areas which falls in private lands. So AMD, that is going to stand for acid mine drainage. And here you see, like I have highlighted 5 locations, A, B, C, D, and E. They represent different topography in this area. So D is that the yellow color that you saw, that kind of a ditch that you can see in this area. This is a close-up where you still have a lot of mine findings. So there are people who try to actually get these mine findings and refine, like purify and sell them. And this is a close-up area of that ditch, which is here, side D. And you see that how yellow is the water here. So we collected soil samples from 5 of these sites and over 2 seasons, fall and summer. And we gathered about like 60 isolates. They, on the plates, they look like different morphology. So we try to recover them. Now I'll not go into details of microbiological methods that we applied, but just to tell you, like these isolates were mainly recovered in the neutral media, which is a nutrient-rich media. But we also try to see that some of these, if these isolates has kind of tolerance to different pH. So we were able to isolate some of the acidophiles, which are like acid-loving bacteria. Now, Rachel did most of the microbiological work, and we found some characteristics like gram-positive, negative. It's because of the gram staining. We differentiate them, and then we also confirm the IDs with growing them in differential media. It's a lot of work, and then we run a panel of biochemical tests for characterization. And you see nice colorations comes out when you do all these biochemical tests. And this is a table that represents like how different these bacteria were from each other based on biochemical test. So we got very like a high metabolic diversity from that area. And this is just 10 representatives, and we chose these because of higher diversity. But all the rest were also having quite unique properties. Now, these are called phenotypic and biochemical characterization. We did a little bit of genotypic characterization also, but it's only based on one gene. So we wanted to identify all the bacteria, not only based on biochemical test, but we wanted to add at least one gene, which is called 16S rRNA gene, unique to the prokaryotes. We use that to create a phylogenetic tree, okay? And we saw like the bacteria that we obtained, all those 60 isolates, they belong to either Firmicutes, which was the majority, actinobacteria and proteobacteria. Among the proteobacteria, we mainly came across gamma bacteria like Pseudomonas pantua, like that. Now, why we want to apply bioinformatics or whole genome sequencing basically in this particular project, because whatever characteristics we did microbiologically, that was good enough for a thesis, and Rachel's work published as a thesis in that way. And then Brady, when he came, we added this component to the same project. The main reason is, like, if you want to have a super degrader, then there are many studies who does like genetically manipulate the degrading stains. Now we are at an advantage because we are really isolating the indigenous microbes from that area. So they have the capability to grow in that adverse condition. So if we can make consortia and also genetically manipulate them, then the bioremediation or clinical projects can be like expedited. Application acidophiles, this part we didn't touch much because we need a sulfate reducer also, because we are really considering to increase that pH of that acid mine drainage area and then make it suitable for growth of the aquatic flora and fauna in that area. So, but we didn't really focus on that at this point. So for whole genome sequencing, it is next to impossible to sequence all those 60 organisms, right? So we reduced the number, only the ones that showed most like metabolic diversity. We tried to sequence those, and then whatever sequence we are going to get, it's going to be an asset so that we can look into their genes and select the ones or combine the ones that are going to be suitable to clean up that site. Now, although I'm talking about like the sequencing cost has decreased a lot, but still there is, you have to put some of your fund to the sequencing thing. So here is an overview, and we are fortunate actually to have a collaborator. In the beginning, I mentioned about K-INBRE. So we have these INBRE collaborators throughout the states. We have one collaborator in New Hampshire, and they helped us in that sequencing. So we were in that way fortunate and to sequence all these 10 isolates there. And we kept doing that in other projects also. Whatever isolates we get, we generally collaborate with them. So the overview is, you first extract the DNA, and then you quantitate it, and then you send for sequencing if you do not have your own facility. Once the sequencing is done, the assembly is going to come back to you, and then you're going to annotate. So assembly and annotate, these are the two things that were new to us, and we learned with help of tutorials and with attending workshops and things like that. So we are now capable of doing it in our lab. You need to extract pure DNA, so that's why we do not go for other methods, but it's better to use some kits. The Sigma kit works very good. And of course, the quality is very important. We realize like it is not really about the quantity of the DNA that you're getting out of the microbes, because some of these, among these 10 microbes, also some of them were really difficult to grow and difficult to extract the DNA, means grow enough to extract DNA. But to get good quality of sequence, if you have a concentration, which is about like, for, I'm talking about Illumina, if it's between like 25 to 50 nanogram per microliter concentration, and that should be good enough, but it has to be a very high quality. Then only you'll have a good sequence. So we were able to get that and send that to Hubbard Center of Genomics in that New Hampshire. And they use this Illumina MESEC sequencer. And then we retrieved the data in the RON server and used Linux command line to learn all these assembly and annotation things. The PuTTY is a small like outlet that we used in the lab, in our site, to access the data from the RON server. So sequencing is done, and then we used quite a few different programs for systemomatic and concatenation. Then we assembled by using spades, and then the quality of genome that also need to be assessed. For that we used QUAST. And then we went into genome annotation. If you're dealing with prokaryotes, like the bacterial strains we have, then we used PROCA. And if you're using eukaryotes, then you have to go for some other program. And we had set of genes that we really wanted to look at. So the targeted genes were looked at. And for identification purpose, we retrieved 16S rRNA gene and translation initiation factor 2. So these two genes were used to identify the organism using BLAST. So when in the Hubbard Sequencing Center, once they are done with the sequencing, the first thing we got is this statistics. And in the statistics table, there are a lot of things. One thing is the quality score, which is a mean quality score. If it's above 30, it is considered good. And I was told that it is good, but I didn't have much knowledge to understand what exactly the algorithm they follow to find out this particular 30 as a cutoff. But when I looked at this table, I saw everything is above 30, so I was happy to see that. Index is something like a tag that you attach to your samples. So those are extra things along with the primer sequence, because Illumina, you have to do some PCR, amplify with PCR, right? So these tags and primers, they need to be removed, and that we did with Shimomatic. Number of reads, the higher is better. It's basically overlapping sequences of DNA that put together. And we are talking about deep sequencing here. That means we try to see unique reads, okay? Number of unique reads, that if the number is increased, then only it's considered deep sequencing. And we got a coverage between like 30 to 50x, which is considered again good. So all these parameters that I'm talking about, I really relied on other like bioinformaticians who gave me this kind of cutoff numbers or ideas, so that I didn't really have to take a bioinformatics course as such. I'm not going to go over all other things, but these are certain things that are very important to look at when we get back this statistical panel. In terms of catenation means, we are talking about again PCR in that Illumina sequencer. So we get forward and reverse sequences. So first thing you are concatenating means joining them together. And then Shimomatic is the program that actually helps in removing the barcodes, primers, and if there are some bad sequences, they're going to just trim it. So trimming is done by the Shimomatic, and then they're also going to pair these forward and reverse for individual sequences. Spades is the one that create assemblies based on these smaller sequences. So the role of this particular algorithm is to join the overlapping regions and make larger contigs. Contigs, the word is coming from contiguous sequence. So you have contiguous stretches of sequences, longer assemblies now. Now, once you have this, after that, we generated some reports called QUEST and PROCA report. So QUEST stands for Quality Assessment Tool, and then PROCA is for Prokaryotic Gene Annotation Tool. So quality assessment, again, once you have all these, like the quality score high enough, it's going to give you some results. In this QUEST report, we generated number of contigs, how many you have. The larger the contigs are, it's better, of course. So the largest contig length also you can get. So the whole genome size of bacteria is around what? Can you guess, like, what is the size of whole genome of like E. coli, probably? Is it in like thousands, millions, or billions? See the chat here. Very close. It's in millions. Thank you. 4.6, and then you get millions of bases, whereas in human genomes in trillions, right? So millions of bases, and what numbers you're seeing here in terms of contigs and total length, they are all in millions. So we are dealing with environmental bacteria, and they really have large kind of larger genome size if you compare them with the parasitic ones that are inside host or cause diseases. They may be smaller. Now, some pathogens may have bigger ones because they have all those extra like pathogenic islands. But in general, if you compare environmental ones, then they are meant to deal with environmental stress. They have many additional components there. Okay. So what we got that corroborated with what we know about the environmental microbes. So these are not aberrant numbers. G plus C content, this is helps to determine the thermostability. So again, in the environment, we see most of these formicutes and actinobacteria, they have higher GC content. And the range that you see here from 38 to 72. So formicutes has a bit higher range. So that is again telling us like the diversity of different organisms among only these 10. So if you consider all 60 organisms that we dealt with, it's even higher. Number of RNA copies also varies. So you can retrieve these RNA copies here. You see it's from 6 to 19. So again, some diversity here. So we are really dealing with different types of bacteria here. And then the coding sequences that will tell you something about how many protein molecules there or open reading frames are present. So it's in thousands, between 4 to 6,000. And then there are some other information that you can get. So these are good things to know about your organism before you deal with them in future studies. And again, this is not only, in the end, I'll talk about this, like this is not only that I implemented in my project, but this I made kind of a component of one of the courses I developed over these years. It's an upper level biology course. So we teach also these things to our undergrads so that they can have some takeaway once they are done with their biology major. They'll know something about bioinformatics. And it was good to have this first-hand data because we did it in the lab and then we took it to the classroom and analyzed it with the students. Environmental ones, I already discussed that. So now let's talk about the genes of interest, because probably the bioinformaticians, they are going to look at these assembly parameters and like the quality, everything. But when it comes to the genes of interest, then you have to really consult a biologist, because depending on what area you are going to implement the work, you need to know that what are the genes that are going to be important. Okay. And that is why it is an interdisciplinary area, because you need to talk to each other. It's not something like you can do all by your own. Probably you can, if you're a very smart person, you can just refer to articles and reviews and you can come up with the gene of interest, but it may take time. So it's better to like crosstalk among the scientific community. So since our isolates are the ones that were from environment and the project is aiming to environmental cleanup. So we were really interested in this set of genes, which we know that have implementation in bioremediation. In my PhD, it was all in environmental microbiology, and we are looking at different genes. So it was easy for me to go into this and collect the set of genes. I'll just give you an example of monooxygenase and dioxygenases that are one of the like important genes to consider when you are looking at a degrader. They can transform many contaminants like organophosphates, aromatic compounds, and they can introduce this. These are very unique enzymes. These you may not find in like a pathogen that is residing in your body. They have some other purpose. But in the environment, when these smaller microbes are in adverse condition, they are in a pesticide contaminated area or some oil spillage area, they activate these monooxygenases and dioxygenases to take care of those aromatic rings. And that's a critical step for the remediation, because if you do not break those rings, you cannot really metabolize those contaminants. And then we were also interested, since it's a like coal mining area, so there are always chance of having other ores around. And as I mentioned, like in a little bit south, we had this lead zinc mining going on also. So we were also interested to see some genes related to lead, some heavy metals like other than lead, like arsenic, and of course some other metabolic genes. This is a command line that we use. This is one of several that we used, and this may not be complex for the computer scientist, but for a biologist, it was again a steep learning curve, because if you make a single mistake in not in typing this, but just having the spaces, you are not going to get it. The result, this nice colorful thing, is not going to show up. So it was sometimes frustrating, but at the same time, once you get this list, it makes your life so easy. You are not really going to look into individual organisms. It's just a common, and that's going to give you all these important things. So that's how we retrieve all these different genes from the sequences. And then we develop this kind of pie chart. All these designations that you see here, they refer to like summer isolates and the fall isolates, and different genes are distributed. And we try to show you like arsenic, stress genes, they were present in this. And this is important here because arsenic resistance is also not very common, but we can apply these isolates in combination with some others to clean up some arsenic contamination, contaminated areas. I know in groundwater contamination with arsenic is a major issue, and that's kind of, it's not really like human introduced. Sometimes there are areas which have those heavy metals and you cannot do much. Prime example is in Bangladesh. They have a lot of issues with arsenic contaminated water. Here in states also, there are a few states, they have like up in Wisconsin, they have these issues. So they try to rely as much as possible, not on chemical remediation processes, but on these microbiological things. This table shows how the genes are distributed, and transferase genes is one of the major genes, and that's of no surprise because they are the ones, they actually transfer the specific functional groups like methyl glycosyl from one molecule to another molecule. So it's a very important group in the metabolic pathway, and we have most of them here. We get a few of the lead genes, which was interesting. So again, they have some implementation. This particular organism might have some implementation when we are going to look at the tarcric side. And so based on all these abundances of the genes, what we conclude here, like substantial diversity in terms of genes in these 10 isolates. And at least one gene that we looked at were actually positive, and we got some bonus, like the lead genes were present, arsenic genes were present, which was really good news for us. And manganese we didn't find in one, but the rest had some manganese related genes. Transferases, as I mentioned already, it's no surprise, but stress genes, very important because those are the ones that will help in surviving like desiccation, high heat, different pH conditions. So they were definitely very important. So this is the first like genome announcement that we had from our lab, and all these draft genomes were submitted to the NCBI. They were reviewed by the professionals in that who maintain those databases, and then we got this publicly available. And not only that, when we did this genome sequence, that actually not only opened up this kind of publication opportunities, but another offshoot, like xenobiotic degrading enzymes. Xenobiotic means, again, it is like pesticides. They're like foreign enzyme compounds. These enzymes are known to be residing on mobile genetic elements, like the different kinds of mobile genetic elements like plasmids, integrons, we have transposons. So it will be really nice if we can look into these mobile genetic elements present in these 10 sequence organisms. Okay, and then once we see like the plasmids are present and they are carrying this kind of genes, those organisms, we can take out the plasmid from them and then we can introduce that to our already known super degrader like Pseudomonas fudida. It already has got several genes. So we can introduce those plasmids. So this is another offshoot, you can say, like that came out of the project. So now we are trying to learn these programs like Plasmid Spades, Plasmid Seeker, Plancet, PlankNet, to analyze the plasmids that are present. So far we found like among these 10, only 4 of them has got some plasmids which significant size. There are some plasmids which are not very big, but the xenobiotic degrading enzymes, they are generally residing on bigger plasmids, which are going to be in like kilobases or sometimes in MBs also. And then we are going to characterize them. So this is going to be another project for a master student to look into the plasmids. So it really helped the lab to expand in the field of this bioinformatics. This is the like take home. Like if you have more catabolic plasmids, then it's going to be a better candidate. So that's the logic behind this particular branch of this study. I don't have much time to cover, but I would like to just introduce, like, we didn't only apply bioinformatics or whole genome sequencing in the bioremediation study, but in food safety, FDA, they are using a lot of genomics to analyze or to decipher the source of any foodborne illnesses. So they take out the organisms that are causing an outbreak, and then if the outbreak is spreading over different states, they are going to have samples and do a quick genome sequence and try to compare. So it's comparative genomics, and that way they can tell like what is the source. So in food safety, there is a lot of application, and that's how I introduced this in one of my project, which is a collaborative project. And Elena was the one here. She alone developed the skills of microbiology. So she dealt with all these poultry feed isolates and also learned the bioinformatics and applied that. So she really did a good job. And now she's pursuing a PhD in food science in University of Wisconsin. And the previous student, Rachel, also, she's pursuing a PhD in University of Arkansas in soil-related program. So these are certain things that as a mentor makes me happy, like the skill set that they are getting from doing these projects, they have applications in their future academia. So Elena's project also led to this sequencing of 11 bacterial species, which are very unique to the poultry feed. And when we talk about food safety, it's not really looking at only the contaminants or foodborne pathogens, but here we are following a different approach to look at something called hygienic indicator bacteria. Because when you're looking at different steps of this food safety pipeline, if you're getting the contaminants in the end, so at the same time, there are some microbes which are going to be present along with the contaminants. So we came across 2 of these, Enterococcus gallinarum, which is a common organism in the poultry feed. It's commensal to the poultry also. We mainly looked at chicken microbiota actually here. And then Pantoa vagans, which is a plant epiphyte, and it's an environmental friendly biofertilizer. So these 2 organisms we proposed as hygienic indicator bacteria in the food safety pipeline. Along with that, we got some pathogens like Cosaconea, Klebsiella. They are known pathogens, human pathogens that can cause blood-borne diseases. Stenotrophomonas, this is a gram-negative bacteria, is opportunistic pathogen. So we came up with all these, but eventually we were able to identify some of the hygienic bacteria. This is a table from the same paper, and this many of you have submitted to Genome Announcement. So you know that this is a comprehensive table that shows like how you have your assembly metrics and annotated features available to the public. I think this is all about my research. So I want to now again emphasize on the impact of informatics to microbiological. So major impact I see here is on students, both undergrad and the graduate students. They really develop this core skill, and doing all this research, they have now expertise in microbiology, molecular biology, and bioinformatics. And as I said, like they're pursuing PhD programs in research institutes, which really makes us happy and gives a good reputation to our university. I developed this microbial physiology upper level course, and have this genome sequences as a hands-on, as a core component of the course. So that way we are like delivering something good to the departmental curriculum, which is again going to be very helpful to flourish, because this is the future. And of course, as a faculty, I am getting some bioinformatics scholarship grants and like publications. So that's going to grow me as a faculty, of course. So it's like a tri-pronged impact that you can see, and it's very well received. I would like to acknowledge my students, both graduate students and undergrads, especially the 3 students that I presented today. And we have a very collegial department. All the faculties are very supportive, especially introducing a new course with changes. It's sometimes difficult because you have to go through different steps for legislation purpose, and all the faculties are really supportive in this. And so I was able to implement this whole genome sequencing in my microbial physiology course. I'd like to thank my collaborator. He has moved to now University of Wisconsin also. And then Kelly Thomas, he was our K-INBRE, INBRE collaborator, and he helped me in getting the sequences done in his facility. With this, I'll conclude. That's my favorite bacteria. I spend a lot of time with them. Anybody can guess this? What it is? It's on a selective media. I know there will be some microbiologists there. Let me give you a hint. This particular bacteria shows 2 different shapes in its like whole life. Sometimes it's a rod and sometimes it transforms in cocci. And it resides in the gut. Enterococcus. With that, I'll just conclude. Thank you.
Wow, what a talk. Thank you very much.
Should I stop share the screen?
Yeah. Please. Yeah, that is fine. Yeah. Oh, Anuradha, what a detailed discussion talk. This is so good.
Thank you.
Can you? Yep, this is so good. Actually, you know, what we are doing here in JIS IASR is highly in line to your research. Maybe not the projects, because we don't have Kansas coal mine here, or we are not working on the poultry farm. But actually, bacteria are bacteria, whether this is opportunistic or the good bacteria. So here also, yeah. This is very much similar-ish that we are interested here in.
Really, all my pleasure to talk to you about my project. There's a very little thing that we do here, but it's always a good idea to just share it.
No. This is so good. This is so good, and you discuss it so nicely. Yeah. So, like many others, for example, like many other scientific lab, we are also working here in coronavirus. So, but the good thing is, in addition to our dry research lab, we are also here planning to get some strains from hospitals, and actually we are fortunate to have our own Illumina MySac sequencer.
Yes, I saw that.
Yeah. And I also saw that you are also an expert on the Illumina.
Well, I'm not expert on sequencing. The Illumina part, I don't know really much.
Yeah. Okay, actually, I was thinking to organize a joint hands-on, so that will help our students. So from our
Definitely.
side, we have an expert, and you are also expert. So we can or combinedly we can organize a workshop kind of things or
That
virtual
will be,
lab
that's
kind of
really a good
things.
idea.
So
Mhm.
yeah. So that would be really great. Okay, I will talk to our department and. Then we can jointly organize another webinar type of things. What do you
Mhm.
think? Okay,
Sure.
okay.
Sure.
Yeah. So I have a question. So, not the scientific question, but some other question. That is, first of all, what do you think? Can we collaborate? Are you interested to work with
Of course.
us in Kolkata? I know you often visit, so we can do some
Yeah.
intellectual
The visiting
exchanging.
part may not be often, but I'm very open to any kind of collaboration.
Exactly. That is really good. And actually, here we are collaborating nationwide as well as with institutes and universities here in JISR. Our main focus is we are encourage we usually encourage our students to be a part of such collaborating projects as their summer project to train
Mhm.
them. As a teacher, our main aim is trained our students first of all to fulfill their degree successfully, and then train them as an independent researcher so that they can
Yes.
pursue their PhD. And
Yes.
yeah, this is our only as a teacher we get
Uh-huh.
only our satisfaction if we see
Exactly.
our student
That's
is
the
in a
reward,
good position.
actually.
Yeah,
Yeah.
that is our reward. And also our aim is to train them so that they can have now the job prospect is worldwide is not that very good. So we try to train our students so that they can have good training in this
Correct.
yes aspect. So yeah, so with your help, so we can collaborate, and at the same time we can complement each other. So
Of
here
course.
the bioinformaticians that from our institute and the experienced microbiologists that you are, so we can collaborate and we can complement
Yeah.
each other.
When I see your faculty profile also, I think like it would be a great idea to have guest speakers. We do have seminar series. So if we can have some of you as guest
Yes,
speakers
yes.
online, that will be great.
Okay, yeah, that is really great, really great. So let's start the question answer session, I guess.
Mhm.
You can also have the access in the question and answer Q&A, right?
Oh, yes, I got it.
Yeah.
Yeah.
So yeah, from Peter Van Hudson,
Houston.
he asked what what you describe can be done quite well with Galaxy, including on the public servers. And he wrote the name of the public servers:
Mhm.
use galaxy.eu, use
Mhm.
galaxy.org,
Yeah.
and use galaxy.org.au. Is there any reason you choose command line over Galaxy?
Actually, I came across Galaxy later. When I was going over these tutorials, I didn't attend a workshop, and in that workshop later, actually, I came across Galaxy. So in future, and the workshop was not attended by me. Elena Olson, she did attend that, but she praised about Galaxy, and that is something probably we are going to use because definitely the error rate in the command line use is going to be less if we use Galaxy. So thanks for suggesting that or reminding me, because that's something I may not use for my like the class that I teach. For that hands-on, I think it's better to give them more like common-based training, so that they are not if they need to do that. But for my research purpose, I'll switch to this, and um, it will be beneficial for us. Thank you.
Another question is from um, Shumanti Gupta.
Mhm.
Did you find any conservative features between those diverse group of bacteria?
So conservative features, actually, we use a program called Orthogene Finder. That's one thing that gives us a set of genes which are common among these 10 isolates, and another set which is not common, that is unique. So in this case, actually, when we use the ortho finder, we were more interested in seeing the unique features. So we really didn't pay much attention to the conserved features because we thought like that's more like the common housekeeping ones that we are going to see. But the unique features we got, and those bacteria with those unique features, with some specific genes, those are the ones we are planning to combine to make the consortia. Okay, so that they can actually complement each other. So again, this part we didn't discuss in the result, but we did undergo that, we use that orthofinder and try to compare. So it's, you can say it's a comparative genomics, not a full-fledged comparative genomics, but partly our aim was to find really the unique features in this case. Thanks for the question.
Another question from Smriti. In order to learn the bioinformatics application, should we go to learning in an online course system or learn via some professional?
Well, what we are seeing these days is like everything is available online, but you need to have some kind of guidance, otherwise you may get lost. But that doesn't mean that you have to pay a lot of money to get into a program. There are some platforms, they give like free tutorial, and that's really good enough to have the basics. And then of course, Smriti, if you get in touch with your faculty, they can put you probably close to some professionals to have like a better idea on some particular aspect that you want to learn. But think twice before you really pay a lump sum to get enrolled in a program, because do the basics first on your own and then you get in touch with some professional. In that way, I think I can suggest like that could be a route that you can follow.
Okay, next is from Ritu Parsrija. Ritu asks, the cultures obtained from site cannot be cultured in lab. So which media is used for isolation and enrichment?
Yes, so there are two ways to study this kind of bacterial diversity. One is called culture dependent and the other one is culture independent. Now, when you do culture independent, it's more like DNA based studies, okay? You are not getting hold of the real bacteria in that case. So we didn't follow that approach. We wanted to get real bacteria on the plate so that we can like apply them if it's needed to the site, so back into the site. That's why we follow really the culture dependent one for that. And that is why if the culture primarily is, we are not able to grow using our nutrient rich media, then it is not going to be of our use in this particular study. But there are many studies I know that they focus on enrichments, and we did that in past in our PhD program when we were looking at the pesticide contaminated areas. We were isolating the bacteria from there, and then we were putting that particular pesticide that is found in the site, adding that to the culture so that the culture get used to it, and then we enrich the ones that are able to degrade it. So that's another approach. In this particular one, we didn't want to go for enrichment because we really wanted to look at a broader aspect, getting taking into account all the like native microbiota that is available there. I don't know if it's answering your question directly, but the part that looked at acidophiles, that was kind of an enrichment method we wanted to allow, but we did not have a surface reducer, sulfate reducer. That's why we really couldn't complete that. But we did follow this enrichment, like we were having different pH buffered media and run those soil samples through the media to enrich some acidophiles. That part maybe we can address, but not really for this project, because this project is more about the remediation of the whole site and not only that particular ditch that we saw. Thank you for your question.
Okay, from Shraddha Mahendra. Hello, ma'am. It was a nice presentation. I have a query. Do we have any sort of opportunity for doctoral research in your laboratory?
I do get some inquiries on that, and I feel sorry to turn them down because this is, again, we have only master's program. We don't have a PhD or doctoral here. But if in future I get some grant, like which is a bigger grant, NSF, NIH, or USDA grant, then there I can ask for a postdoctoral position, and that way I can have a postdoc, but I am not that far yet. I have to work on that. So there is no harm in approaching and asking. If I do not have a position, I can always direct you to some other people I know with the same like matching interest, and you can approach them who are in actual research institutes and have more postdoctoral openings. Okay, good luck.
Okay, sorry, Anuradha. She's not asking for postdoctoral position, but she's asking for doctoral research position. So
yeah,
PhD.
I misread that. Yeah, so PhD program also, unfortunately, we do not have here. Actually, we can have postdocs probably because of the grants, but we cannot have PhDs here in our institutes.
Mm-hmm, yeah.
So for PhDs, you have to really apply for other laboratories.
Yeah, yeah. I guess that is she's depressed with your answer, but there is always chances from other chances, other labs, other scopes. So from others, we have another question. Have you had any experience in human gut microbiome research?
Not really. I never dealt with any human samples yet, but I have dealt with some other like food animal samples, large animals like cattle, swine samples also. It's going to be different from human, but may not be too different because we know that their genes are shared quite a bit. But I do not personally have any experience with human gut microbiome.
Okay. So from Shraddha Mahendra again, can you guide me to some other person as I'm from biotechnology field?
Sure. For PhD program, I know people in my previous university and my current collaborators. They're all from like research institutes. So there are opportunities available. So if you can contact me personally by email, I can share whatever I know, and then you can contact them directly. Good luck again.
From Madan Varma.
Mm-hmm.
Working with robust microbes during coal mine, what is your conclusion of their robustness based on their genome sequencing studies?
Very good question. We were amazed to see the number of stress genes that we found. And that's really promising. It's not only stress genes, what we mentioned about the arsenic, heavy metal tolerance, and lead genes, they are like additional ones that we got. So you may not really, although we are looking at environmental, but it's really, the microbes are so interesting and they are so different when you compare different kinds of environments. So in coal mining area, we see this set of microbes with all these tolerance and stress genes. When we are looking at a oil spilling or oil spillage area, there may be completely different set of microbes. So the uniqueness and robustness is definitely something noticeable in this. And we are in the way of communicating a manuscript where we are just focusing on this characterization part, and we are talking more about all these different microbes with their ability to work with different kinds of xenobiotics. So that will be something that we are addressing there. But it's a very good question. Thank you.
From Ritu Freezer, does human gut samples require ethical technical clearance?
As I mentioned, like I'd never worked with it, but I think you need, from my perspectives of handling food animal samples, like again swine, poultry, and cattle, we needed some clearance by the USDA. Okay, so for human, I assume like you have to go through all these IRB and IUCAC and all these different agencies are there. So you need to get some clearance.
Okay. Any more questions? I can't see any more. Anuradha, would you please write down your email ID to the chat section, because there are many requests to contact you directly.
I am really happy to see.
No. Anuradha, there is a problem. I'm sorry. So what you wrote, that is to all panelists. Can you?
Okay, let me just share my screen here. I think I have it on my.
Yeah. That.
Well, I do not have it here. I'm sorry.
So just write it down on the to all panelists and attendees. Okay, let me copy it for you. Okay. So
can you see my screen here? I just wrote it here.
Yes.
Oops.
This is A. Ghosh at
pittstate.edu.
State. Yeah, I wrote it down for you.
I just wanted to mention I'm really impressed to see such a number of participants that you have, and that too on a Sunday. They're giving up their probably family and friend time
okay.
to attend the seminars.
Okay, there is another good input that is from Peter. Yes, that is from, I can't see the name, I'm sorry. Peter Van Hudson. That is for those wanting to learn some basics with Galaxy. Just he shared the website, the link for the tutorials. Yeah, thank you very much for the link. Okay, so any more question? I can't see any more questions, but lots of thank you. Nice talk, ma'am. Very important, very resourceful.
My pleasure.
And request.
So can I take
Dr. Chattopadhyay? Yes.
Yes. Dr. Shuja Chattopadhyay from our institute.
Yeah, is my colleague. Anuradha, it's a great talk, and I have been a privileged attendee so that I can ask the question to you. I don't write the question on the chat section. It's just because of everybody who made me privileged. So a couple of questions. The first one.
Yes.
Did you get a chance to compare the genomes that you sequence from your geographic region, the ones from other mines in the US?
And yeah,
that's one
great question. But we really means we have it available, but we haven't done, I think, enough on this, because that's a definitely good idea, and we would like to follow up that, because there are many other, like in Florida and other parts like Montana, Wyoming, there are coal mines and there have been studies. So it will be really interesting to compare these local microbes with the ones that they have.
Yeah, because when you were talking about this horizontally transferred genes, mobile elements, and then you were looking for those features, but if you compare your genomes with genomes from other part of country, other part of the world, depending on the similarity and difference, you might get very important variations even in the housekeeping genes that might very good inputs, because in many cases housekeeping genes are under positive selection pressures.
Okay.
for many different reasons, and in many cases, those horizontally transferred genes, even if they're there, when they're going to get expressed or not getting expressed, being controlled by these housekeeping genes.
Yes.
So, because there are many such instances, especially in case of what we know for microbial virulence and antibiotic resistance. It's very much true. This clonal structure is very much important. So, and variations are very important.
Yeah,
So,
that's really a very important piece of information. This the variation in housekeeping genes, not for these, but in past actually we were studying that using some genotyping approach like MLST, MLVA, PFGE. So what you are saying is really means perfectly fit in this scenario because we see the variations there in those housekeeping genes.
yeah. So it would be great if we get a chance to collaborate and we can write joint projects.
Yeah, I'll look forward to it.
Hope
And also, do not forget our exchange program.
Oh, yes.
And just one, although the question was asked to you, but there was one question about bioinformatics thing someone asked. I'm trying to find this. Yeah, in order to learn the bioinformatics applications, should we go learning in an online course system or learn via some professional? So can I add something, because you you are the main person here. I mean,
please go ahead. You know more than me on bioinformatics, of course. Please.
No, I mean, I believe you answered it definitely from your perspective, because being a microbiologist, if someone wants to know bioinformatics, I'm absolutely buying what you said. But if someone really wants to be a bioinformatician, someone wants to be a computational biologist, then probably just online tools won't help. If it would help, then worldwide, the courses, the master's courses offered by all these different universities would be totally useless.
I buy your logic completely. Yep.
Depending on, whether you want to make it your priority or you want to make it a kind of an auxiliary.
Exactly. Yes.
Yeah. So,
yeah, probably I answered more from a faculty point of view, and what you're saying is really for the students, so it's going to be helpful to the student.
Yeah. Otherwise you just have to think that we will be jobless, right? We'll be so all the courses, scientific courses all over the world, they will be totally gone. So Sriti, if you are listening, if you want to be a bioinformatics person, then you have to join a bioinformatics course, definitely. But if you want some kind of, because bioinformatics is a vague term, what kind of bioinformatics application you want?
Yeah.
It's all different kind of forms. All different things are there. So you have to find out what kind of applications you're looking for. And as Anuna was saying, she was absolutely right that you have to find your faculty can guide if you want to get some help, and then you can move accordingly. But if you want to make it your profession, then you should have the complete coursework.
Right.
So,
without certification, it cannot become a bioinformatician. Probably right. Yes.
Yeah.
Thank you. Thanks.
So Sriti is thanking you, Dr. Chattopadhyay. She wrote in chat section, "Thank you, Sujoy sir. I am from microbiology background, therefore wanted to gain some insight into the bioinformatics application part that would help me in my research."
Okay, Sriti, then just trash whatever I just told you, just trash this information. So what already Dr. Ghosh informed you, that's the only thing you should look for. Because
Yeah.
you just want to have bioinformatics as a helping guide for your research. So you can get your collaborator, some collaborator who can help you do that, or you yourself can find out depending on the area of bioinformatics.
From Peter Van Hertzen, one more tip, the microbiome file podcast discussion techniques in microbial bioinformatics, and he wrote a link. The hosts are focused on pathogens, but the techniques discussed are quite general, for example, genome assembly or phylogeny.
Yes.
There is too
Yeah,
much
I'm
of
not aware
missing
of this
manual in
particular
bioinformatics.
thing.
Okay.
You can look at.
Okay, yeah. So, so anyway, thank you very much, Anuradha, once again.
Thank you.
Yes, and what I want to add that for anyone who is looking for the recorded talk of Dr. Anuradha Ghosh, email us on the same email ID that you got your link, Zoom link. That means Debobani, DEBABANI @JISIASR.org or biosim@JISR.org. And here I'm sharing that email IDs, and then what we will do, we will send you the YouTube link of the talk, or you may go to the facebook.com https :/facebook.com/ all caps JISIASR. You can find the live of this webinar. That is not live after we end the meeting, but you can have the access, or we can send you the YouTube link, but just email us the request. Other than it is so difficult for us to look for the email IDs from or to find the email IDs of yours from the huge database, because today's registration was 120. So there are lots of attendees. So it's so difficult for us to find the email ID of you. So email us with the request, then we will send you the YouTube link, or you can go to the Facebook, our page, JIS IASR, then you can have that. Or you can email Dr. Ghosh. She can also send you the link. Okay, thank you very much.
Well,
So
thank you, Devavani, once again for this invitation and allowing me to share my work with all of you. I really appreciate it. Thank you.
Thank you. Thank you. So I'm ending the meeting for all. Thank you.